What does a bee's brain actually look like? Well, if you take a bee and carefully slice off the top of its teeny tiny head, this is what you'll see. Just a whole load of white mushiness. But by the magic of histology, we can turn that white mushiness into a colourful array of weaving axons linking neurons. Most of these sections were taken by placing the bee on its back and cutting progressively through the head. Just so you can keep a tab on where the sections are from, I'll include a little diagram in the bottom right corner of the screen so you can visualize which part of the bee's head we're talking about. Bees are colony-living invertebrates, which can demonstrate an array of complex behaviors, such as the famous waggle dance, which communicates the direction and distance of a food source to other forager bees. As invertebrates, both performing the dance and interpreting the conveyed information will be coordinated by a central brain. Sensory information is an important starting point when looking at brain structure. This is the raw information that the animal is gathering about its environment to inform its decisions. Like other insects, bees have a concentration of chemical sensors in their antenna. Each antenna is connected to an antenna lobe within the brain, which receives impulses from the antenna sensory cells and outputs to higher centers in the brain. This is what the antenna lobes look like. They have a very distinctive structure consisting of neurons around the outside and lots of spherical subunits of axons called glomeruli. Each glomerulus serves as a functional processing unit for different odor molecules. There are about 160 glomeruli in each lobe, which receive input from a specific class of olfactory receptor neuron. One of the most important outputs of the antenna lobe is its connection to an area of the brain called the lateral horn, which is responsible for odor valence. In other words, distinguishing between attractive and aversive smells, such as floral scents versus toxic substances. Taste and input from the mouthparts is also an important sense for bees, so much so that an entire area of the brain is dedicated to moving their complex mouthparts and assessing whether what's being eaten is good or not. This all happens in the subesophageal zone of the brain, so called because it's under the esophagus. That's right, this tube going right through the bee's brain is its esophagus. This is a feature common to insects and other arthropods like arachnids. I guess when your diet consists of liquid feed, then you don't need to worry about a particularly large or spiky mouthful causing brain damage as you swallow it. The subesophageal zone is responsible for controlling feeding behaviors, sensory integration from the mouthparts, and the motor functions of the mandibles and proboscis. For example, when the bee's antenna contacts a sugar-containing substance, the bee will reflexively extend her proboscis. This so-called proboscis extension reflex is mediated by neurons in the subesophageal zone. Bees have very complex mouthparts which are involved in a number of social behaviors like food sharing, so it's not really surprised that such a large amount of the brain is dedicated to this specific area. But in terms of the sheer brain area dedicated to it, no other sense comes close to that of sight. Bees have 2 compound eyes and 3 simple eyes called ocelli. Each eye is connected to an optic lobe in the bee's brain, which will process the information and distribute it to higher processing centers. The optic lobes contain 3 regions: the lamina, the medulla, and the lobula. From the eye moving inwards, there's the lamina. Neurons in this area will receive signal from the ommatidia, or the individual eye units, for initial processing. Next in line is the medulla, just behind the lamina. Neurons here receive input from the lamina and connect to the protocerebrum, an area of higher processing. Output is also sent in parallel to the final area, the lobula, which also connects eventually to the protocerebrum. Research on bee visual processing has shown that different physical elements of visual information, such as color, motion, and stimulus timing, are physically separated in the bee's brain. In other words, neurons that respond to color are present in a different layer of the lobula to ones that respond to motion. Segregation of this information is maintained in the protocerebrum, allowing parallel and therefore more efficient processing of visual information before everything is integrated in the higher processing centers of the bee's brain. Now, I've mentioned higher processing centers quite a lot, so let's have a look at them. After all of those sensory distinct lobes of the bee brain, we come to the big pink central mass. At a rather superficial level of histology, we can make out 2 distinct areas called the alpha lobe and the beta lobe. Really, this is a gross oversimplification. Somewhere within this mass there is the central complex, a key structure for motor control and spatial orientation. Essentially, this part of the bee's brain is receiving inputs from different sensory areas and integrating them into movement or behavioral outputs. One fascinating paper describes how bees use the central complex to undertake a process called path integration. This means that they can keep track of distance and direction during a foraging trip to allow them to calculate a direct straight-line route back to the hive, irrespective of how meandering their original route was. The research found that bees can use polarized light from the sky as a compass, and optic flow, in other words, how the environment moves past them, to measure direction and distance respectively. Neurons in the central complex can integrate this information and add up the turns and distances to create a kind of internal GPS that allows them to calculate the best route home. The mother of all integration centers has to be the mushroom bodies. These are located right at the top of the bee's brain. You can see them here as these C-shaped aggregates of white matter surrounded by lots of small neurons. They kind of look like mushrooms if you use your imagination. There are two mushroom bodies on each side of the brain, a lateral one and a median one. The neurons around them are known as Kenyon cells, named after F.C. Kenyon, who described them in 1896. Information from various sensory modalities like sight and smell are integrated here, allowing the bee to process complex environmental information and output an appropriate social behavior or communication. The mushroom bodies are also the center for learning and memory. Bees use their mushroom bodies to remember floral scents, navigate complex environments, and even recognize human faces. The size of a bee's mushroom bodies can actually change depending on its role in the hive. Forager bees, who need to remember flower locations, have larger mushroom bodies than nurse bees, who remain in the hive. Mushroom bodies are not unique to bees, but are found in most other insect species. However, their size, shape, and function vary significantly across different insect taxa, reflecting evolutionary adaptations to different ecological niches. For example, in Drosophila melanogaster, the fruit fly, the mushroom bodies predominantly process olfactory information. They receive input from the antenna lobe and play a central role in odor discrimination and associative learning. Meanwhile, cockroaches exhibit a broader sensory integration role in their mushroom bodies. Studies have shown that their mushroom bodies encode more than just sensory input. They also contribute to decision-making and motor control. Experimental recordings have revealed that when a cockroach perceives food odors, mushroom body neurons actively influence its decision to approach or ignore the stimulus. The evolutionary flexibility of mushroom bodies suggests that their role in cognitive function is not fixed, but rather shaped by selection pressures specific to an insect's lifestyle. The fundamental architecture, Kenyon cells forming calyces and output lobes, remains conserved, but the relative importance of olfactory, visual, or mechanosensory inputs varies widely. But that all seems like a topic for another video and a good place to stop our little tour of the bee's brain. Obviously, it just brushes the surface of neural complexity in these insects, but I hope it was an interesting look at these tiny creatures. Thanks for watching, and until next time, goodbye.